Today we will be covering concept 8.2, alleles and genes interact to produce phenotypes, from Principles of Life, second edition for the AP course, Hillis et al., textbook pages 158 through 162. Now, when we talk about these new alleles, oftentimes they arise by mutations. These are rare, stable, inherited changes in the genetic material. The wild type is the allele present in most of the population. We need to be careful and not use the word dominant allele here. Remember that dominant and recessive refer to different things, but it is the most common one that's there. So a gene with the wild type is present, that is present less than 99% of the time, is called polymorphic. We'll get into explanations in that and examples in just a minute. So one gene may have more than two alleles. Multiple alleles increase the number of possible phenotypes and may show a hierarchy of dominance and heterozygotes. So like coat color in rabbits, okay, for example. So we can see at the top of page 159 in your text in figure 8.9, we see that we have these different rabbits here. We have dark gray, albino all the way over here. We have chinchilla, and then point restricted right here. And as you can see, we have several different possible genotypes for each that can give rise to this, and not just a single color. I know that in the previous lesson, we really simplified things with heterozygote as being one dominant and one recessive allele. But as you can see here, in most cases, we are going to have multiple genotypes, which give rise to this heterozygote being present. So incomplete dominance. Now, sometimes some alleles are neither dominant or recessive, and they have an intermediate. Okay, so the example that we have here is flower color and snapdragon. So instead of being red or white when they combine as heterozygotes, they become pink instead. So the same Mendelian ratios are present in the F2 generation. If we cross two heterozygotes, we end up with that 1 to 2 to 1 ratio like we talked about in the first lesson. If we cross it with a white parent, we end up with a 1-to-1 ratio of pink to white over here. So again, these alleles don't disappear. It's just that they will segregate independently when we go through our F2 cross that's there. Now, codominance is sometimes two alleles of a gene both produce phenotypes in the heterozygote. So the ABO blood group system has three alleles of the gene IA, IB, and IO, where IA and IB are codominant. That is where our AB blood type comes from. Okay, so if we look at figure 811 on page 160 in your text, you can see a table explaining all of this. So you have your four blood types right here. Of course, this is not including the positive or negative, the RH factor in here, but we have the AB, and O blood type. We have the possible genotypes here. Okay, so IAIA or IAIO. We simply need one of those present. However, to be AB, we have to have one A and one B allele. To be O, we have to have two O's that are here. Okay, so epistasis. Now, sometimes phenotypic expression of one gene is influenced by another gene. Okay, so epistasis is incredibly common. So things like coat color in Labrador retrievers, where the allele B, black, is dominant to the allele little b for brown. Okay, then you have pigment deposition. Allele E is dominant to no pigment deposition. So the hair is yellow. Gene E determines the phenotypic expression of gene B. So we can see this on page 161, figure 8-12. So right here we have a dog with alleles big B and big E is black. A dog with alleles little b, and big E is brown. And then this yellow lab over here, a dog with little e is yellow regardless of its big B, little b alleles. Okay, so we can see how these will pair together and give rise to the different ones that are there. So again, we can take two that appear black, two black labs, and when we breed them, we end up with this mix right here. So this is one of the classic examples of epistasis. It's taught all the way from the introduction of genetics in like 8th grade science, used all the way up through college. It's a model system, a lot of, similar to what we talked about with Mendel's pea plants in his courtyard. So sometimes we have hybrids. Now, hybrid vigors or heterosis, this refers to hybrid offspring that can grow larger, produce more seeds than the parental varieties. So corn is the best example of hybrids. And this is on page 160 in your text. We're jumping back for a second. But hybrid corn is very common. This right here is typically the corn that we eat. And then we take these seeds, we grow more corn from it. So we have two different parents that are smaller. You can see that this stalk is slightly longer, but has fewer seeds. This one has more seeds, but is shorter. We hybridize them, and they produce this larger corn stalk, which, the larger the plant can become, the more profitability there is for the farmer. So vigor is a complex trait. Most complex phenotypes are determined by multiple genes. So again, most of them are not going to be determined by a single gene. This is one as well. Most are quantitative traits, meaning they must be measured rather than assessed qualitatively. So grain yields, again, if a farmer can produce more corn, the more corn he produces, the more profitable he is, the more likely he is able to survive. And additionally, the more potential offspring or seeds that a corn plant makes, the higher the likelihood is that they will sprout and go through reproduction themselves the next year. Okay, so genotype and environment often interact to determine phenotype. So we talked about this point restriction in rabbits. Okay, so this is an example right here. We see a Cheshire cat right here, and the same patterning in a rabbit. So when we talk about point restriction, we mean endpoints on the body, so like the nose, the ears, the ends of the paws, tail, all that. They will tend to be darker than the rest of the body. So this is determined by the environment. So those rabbits and cats have a mutant allele for the coat color gene. There's an enzyme that is inactive at temperatures above 35 degrees. So the extremities are cooler than the rest of the body. Then the main body is around 25 C. So the fur on these regions is dark. Okay, so if we expose them to that, that is what we will see. That's the effect that we'll see taking present. Okay, so there's two parameters to describe these, the effects of genes. We have penetrance and expressivity. Penetrance is going to be the proportion of individuals with a certain genotype that show the expected phenotype. So if we expose all of these kittens to the same conditions, what percentage of them have this coat pattern activated, and then expressivity, the degree to which each genotype is expressed in an individual. So is it just right here at the tips, or is it further up? Is it halfway? We can make a measurement of that. Okay, so many people, but not all, who inherit a mutant allele for the BRCA1 gene develop breast cancer. The mutation is said to be incompletely penetrant, and a woman with the mutant allele may get both breast and ovarian cancer, but another woman may only get breast cancer. This mutation is said to have variable expressivity. Okay, so this is a very common test done in the hospital now. It's a blood test. They come in, draw a little bit of blood. They do genetic analysis, and they determine whether you have the mutant for this gene or not. It's used as a genetic screener for a increased likelihood of breast or ovarian cancer. Like I said, it's a common test, and a lot of women in their 30s, early to mid 30s, are starting to have this test done. So we also have heritability. Okay, this is the relative contribution of genetic versus environmental factors, okay, to variation in a character in a population. So the good example of this is height. If we were to measure the entire school, this is what we would generally see as well. We're going to see, this kind of bell-shaped curve here, where we have a few very tall individuals over here and a few very short individuals over here. And this heritability ranges from 0 to 1, as we've already talked about. For human height, heritability varies from about 0.65 to 0.8. Okay, so if the heritability is 0.7, 65% of the variation in height is due to genetic factors, 35% is due to environmental effects. So we're going to talk about this part in class. Think about what the environmental effects could be. And heritability estimates apply only to variation within populations, not individuals. Okay, so we're looking at an entire population, not a single individual. If you have any questions, put it in the comments below. Make sure and bring them to class.